All right, let's just dive right into this. Welcome to today's Explainer, where we're going to bridge that massive gap between hard structural biology and high-end Hollywood visual effects. We're looking at a process happening inside you right this very second, the nitric oxide signaling cascade, and we're going to break down exactly how to visualize it. We're talking absolute scientific accuracy combined with a stunning photorealistic bioluminescent style in glorious 8K resolution. It's like turning raw biochemical data into a cinematic masterpiece. But before we jump into the 3D pipeline itself, we really need to understand our biological star here. We're talking about soluble guanylyl cyclase, or sGC. This thing is a microscopic marvel. It's an obligate heterodimer enzyme, basically a two-part molecular engine, and a totally intracellular gasoreceptor, floating right there in the cytoplasm. So what's its main job? It waits to bind to a gas called nitric oxide to produce cGMP. And cGMP is that exact chemical messenger that tells your smooth muscles to relax, causing vasodilation. Honestly, without this specific enzyme doing its thing, our cardiovascular systems would just stop working. So here's our roadmap for today. First, we'll cover the nitric oxide signaling axis. Second, importing that PDB's structural data. Number 3, animating those molecular state transitions. 4, simulating the cellular fluid dynamics. And finally, number 5, bringing it all together with cinematic rendering using Octane. All right, kicking things off with part 1, the nitric oxide signaling axis, deep inside the cell cytoplasm. To start off our animation, we have this precise biological trigger. And I want to be clear, we aren't just making things glow for the sake of looking cool. That glowing nitric oxide molecule is rapidly diffusing through the cellular fluid until it finds a highly specific docking station. It's looking for the Fe2 heme located within the HNOx domain of the sGC beta-1 subunit. When that tiny gas molecule finally latches onto the iron in that heme, boom, it kicks off a massive conformational change in the entire enzyme. And, biochemistry literature reminds us constantly that nitric oxide is an absolutely essential signaling molecule in biological systems. This isn't just a pretty visual, right? This exact interaction is fundamental for life. When NO is synthesized from L-arginine, it rapidly diffuses across cell membranes and binds to that heme cofactor of sGC. Just this one single binding event leads to a several hundredfold increase in cGMP synthesis. It's literally the biochemical equivalent of flipping a tiny switch and turning on a massive floodlight inside the cell. Moving right along to part 2, importing PDB structural data, or basically how we bridge that raw data to art. Because we don't just guess what these proteins look like, right? We actually pull their exact atomic coordinates straight from the RCSB protein databank. Researchers have deposited some incredibly high-resolution data in there, like the 2.9 angstrom resolution crystal structure of a cGMP-bound kinase. So we grab that. Then, using an incredibly powerful tool called the Molecular Nodes add-on in Blender, we import those PDB files directly. This add-on maps the atomic structures to a vertex-based geometry nodes mesh. Basically, it turns cold, hard spreadsheets of data into manipulable 3D geometry right before our eyes. Now, to animate this process accurately, we have to compare the precise structural coordinates of sGC in its resting state, which is cataloged as 8HBE, against its fully active state, 8HBH. When that nitric oxide molecule hits the heme domain, the entire protein shifts from state 1 to state 2. But here's the catch. Getting a 3D program to smoothly transition between two massive, dense datasets of atoms, yeah, that's where things get incredibly tricky. Which brings us to part 3, animating molecular state transitions by interpolating those coordinates. So the big issue here is that we can't just use standard 3D morphing. If you try a simple mesh-level morph between different protein conformations, you end up with massive vertex scrambling. The connectivity indexes between the two imported states just don't match up, so the protein basically turns into this jagged, exploding mess of spikes. Nobody wants that. Instead, using geometry nodes, our structural state transitions are driven by direct atom coordinate interpolation using the Animate Frames node. We are literally telling the software, hey, interpolate the exact spatial coordinates of each specific carbon alpha atom from state A to state B. This maintains absolute, 100% scientific accuracy. And to make it look physically realistic, we use an animation parameter, an S-curve, moving between 0 and 1. By checking the smoother step option in Blender, this S-curve perfectly models the real physical acceleration and deceleration of the protein domains as they snap into their active positions. It gives the molecule a real sense of weight, a kind of microscopic snap, rather than just a linear, robotic, fake-looking slide. All right, on to part 4, simulating cellular fluid dynamics, going from GTP to cGMP. So, remember, the cytoplasm isn't just empty space. It is a crowded, chaotic, highly viscous environment. The diffusion of our substrate GTP within this cytosol is governed by a Langevin equation. Inside Blender, we replicate this using a high-frequency noise vector to generate a pseudo-random displacement vector. We add that to the velocity of each particle, and it perfectly simulates that jittery, random Brownian motion. Once the GTP randomly collides with the newly active sGC enzyme, boom, we trigger a real-time catalytic transformation. That particle instantly turns into our vibrant, cyan-glowing cGMP molecule. But, we don't want these molecules to just look like hard, bouncy little spheres. They need to look like actual biological fluids. To achieve this viscous fluid interaction, we procedurally generate surface tension using a very specific workflow. We take mesh to volume and volume to mesh nodes. By keeping the voxel size super small and dialing in the threshold parameters, those individual particles just melt and merge into smooth organic fluid streams. Finally, we slap on a geometry nodes modifier to project the surface normals onto nearby boundaries, simulating actual physical surface tension and fluid-solid interactions. It is literally pure mathematical beauty, creating organic life on screen. And that takes us to our final step, part 5, cinematic rendering with Octane to achieve that photorealistic bioluminescence. So how do we make proteins actually look real? Well, we set our specular transmission material to an index of refraction, or IOR, of exactly 1.35. Now that is not a random number. That matches the actual real-world optical density of typical intracellular protein tissue. By combining this 1.35 IOR with a random walk subsurface scattering medium, light physically bounces around inside the protein geometry before exiting. This gives the sGC enzyme that dense, translucent, water-heavy look that is absolutely crucial for a photorealistic bioluminescent style. It's exactly what you'd expect to see under some impossibly powerful future microscope. And a huge tip here, we can't just light this with standard virtual lamps. The environment is the lighting. We immerse the entire scene in an atmospheric fog. We apply a specular material to a bounding box enclosing the whole camera path, enable an active scattering medium, set the density way down to 0.02, and configure the scattering color to a deep, dark teal. This perfectly simulates the dense background of cellular fluid. So when that glowing NO molecule and those newly formed cyan cGMP molecules start emitting light, their photons physically interact with this fog volume, scattering and absorbing to create these breathtaking, cinematic, volumetric spotlights. But there is a catch when you're rendering something this complex at a massive 8K resolution. To ensure the final render stays totally flawless and avoids any of those ugly, banded artifacts in that dense fog, we have to manually adjust the ray marching algorithm. We drop the volume step length from its default of 4 meters all the way down to just 0.1 meters. This aligns the precision of the render engine with the literal micrometer scale of our molecular structures, guaranteeing an incredibly smooth, crisp, and clean final image. Looking at all the incredible techniques we've covered today, it really leaves you wondering, doesn't it? I mean, if we can actually see the invisible mechanics of our cells this clearly, utilizing exact PDB coordinates and physically accurate light transport, what other medical mysteries are we about to unlock next? Just imagine learning biology not from some static textbook, but from a fully immersive 8K environment where you literally watch drug molecules bind to specific protein domains in real time. It completely reshapes how we understand the fundamental building blocks of life. Think about that as you continue to explore the fascinating world of structural biology and 3D visualization. Thanks for joining me on this explainer, and keep learning.